This is a production of Cornell University.
Okay. Hello, everyone. I'm Ming-Yi. Today, I'm going to talk to you about vineyard under vine soil management. And I'm going to look at it from two perspectives, including a vine physiological perspective, as well as vineyard soil microbial, grade microbial, and wine fermentation microbial perspective. So if you look at the picture, you will notice that bare soil under vine for both the picture. So maintaining bare soil under vine is the most popular vineyard management practice in our region. And the reason for that is we basically inherited this from the juice grape industry, where they needed a larger vine to support higher yield. But for wine grape industry, this is excessive vigor is a problem. People are just sending their crews to do hedging multiple times, to do leaf pulling multiple times, just to control the vine vigor. So you know that this is not a very favorable kind of management practice in our wine grape industry. And plus, there's a research done by our group by Mr. Adam Carroll, who is not here today, who found that using glyphosate to maintain bare soil under vine in vineyard for long term, you increase the amount of nutrient leaching and also the pesticide leaching in the soil leachate. So overall speaking, this is not really suitable for a lot of the wine grape vineyard, and we are looking for a good alternative. So many people studied that using undervine cover crops as an alternative, and they found that undervine cover crop can reduce vine vigor through water and nutrient competition. But those studies were done in young vineyard. So what happened in a mature vineyard with those vines with well-established fruit system? So a study done by our group again. In a mature Riesling vineyard, we found that undervine cover crop did not impact on vine growth, did not impact on vine water nutrient status, and did not impact on berry composition. But what we found is that wine sensory properties were impacted, and that really make us think, because if we don't get the effect from the physiological perspective, what else can play the trick there? And so we start getting curious about the microbes. So maybe microbes is playing the trick there. So we want to study more to focus on the microbial perspective in the vineyard. So these are our objectives. We want to evaluate how undervine cover crops would affect on the vegetative growth, yield, fruit composition of a mature Riesling vineyard and Finger Lakes. And we want to determine how soil health will be impacted by adopting different undervine flora management practice. So these are considered as the physiological perspectives. So we also want to know the microbial perspective. So we want to see how flora management practice would affect soil, grape, and also wine fermenting microbiome. And finally, we want to evaluate wine, the sensory property of the wine, to see if the wine sensory property are being affected. If so, is it going through physiology route, or is it going through a microbial route? So our experiment was conducted at Ovid, New York, sitting on the Howard Gravely loam with long-term average growth degree days around 1,376 based on degrees Celsius. And the vineyard was conventionally managed. The vines were mature vines. So in 2014, they were around 14 to 15 years old. And the vines were Scott Henry trained to 16 buds per meter. And these are the treatments we use. So GLY means glyphosate. So we spray glyphosate 1 to 2 times per season, depending on the weed pressure. And we also consider this as our control. The other two treatments are natural vegetation. So we abbreviate that as NV, which you basically just let weeds to grow on their vine. And we mow them down whenever it's interfering the fruiting zone. And CULT, which means soil cultivation. So we did it by using rototiller and hand cultivation. For the whole experimental setup, we adopt randomized complete block design with each of the treatment replicated for 4 times. For each of our experimental units, it contains 9 panels in 3 rows, as you can see here. For each of the panel contains 3 vines, so there's 9 panels. We only collect our sample from the middle panel, which is our data panel, because we're doing microbial work here. So we really want to have that, that physical and spatial barrier to prevent the intervene coming from outside of our experimental unit. So that's why we set up like this, and this, so to be big enough to have those buffer. So let's look at the results first, the physiological results. So vegetative growth, we measured primary shoot growth and lateral shoot growth throughout the growing season for each of the year for 2015 and 16. Just remind you that the experiment was set up in 2014, but we only collect the yield and grape data in 2014. And the growth, like vegetative growth, we only collected data in 2015 and 2016. So it was assessed in these 2 years. So the vegetative growth, we use primary and lateral shoot growth as an indicator. And as you can see here, for lateral shoot growth, we measure shoot length and also the proportion of shoot that had the lateral shoot. And basically, we did not see any difference among the treatments. Just to remind you that at our site, our vines are, the vines are huge and they're mature. So they had that really extensive, really well-established root system. So they got access to those really deep soil, water, and nutrients. So it is very reasonable to not see any difference among the treatment here in terms of the vegetative growth. And we use midday stem water potential as an indicator for the vine water status. So it's measured as bars. And we did not see any difference among the treatment in 2016. But in the late season 2015, I found that natural vegetation and cultivation, basically, the vines in those 2 treatments had a higher stem water potential compared to that of glyphosate. So the theory behind that is, for those 2 treatments, they basically have soil surface protection. So, and in native vegetation in 2015, around August, we had very low rainfall, and then a lot of the weeds are stunt growth. So they've been just hanging out there, but they're not doing like a very aggressive growing. And for cultivation, after we do the cultivation, the plant residues are just left on top of the soil. So those stuff just serve as a surface cover, like a mulch, and they protect the surface soil water to evaporate. So that means for those vines, they got more of the surface soil water. For vine nutrient status, we took leaf petioles at Riesling for each of the year. We did not see any difference among treatment in 2016, but in 2015, we found that natural vegetation basically increased the amount of the aluminum in the petioles compared to glyphosate. So my theory behind this is, for those, if you input a lot of organic matters, and if there's the weeds actively growing in your soil, that means in a very small scale localized soil, there'd be soil acidification, and that freed up a lot of the free aluminum to the plant. So that's why we see this, and that's the theory that I formed. And for natural vegetation, that also reduced the amount of maintenance in the vine by 28% compared to cultivation. And this can just purely due to the competition. And we also collect the vine yield and pruning weight. So vine yield, it's very simple. It's just how much of the fruits for each vine we got. So we measure the amount of fruits for each vine, and we weigh it. That's how we measured the reproductive growth for the vine. And pruning weight. So I'm going to explain you pruning weight. So in each of the growing season, after we harvest and after the leaf fall, we prune the vine into the same shape with same numbers of shoots and same number of buds. So just maintain it into the same shape. And those shoots got pruned off, we consider as the shoots of the concurrent year growth. So we weigh those shoots as our pruning weight. So as you can see here, those are the shoots got pruned off, and this guy's having that backpack weighing scale, and we just weigh the amount of shoots we prune from the vine. So that is the indicator for the vegetative growth for each of the vine. So now we got the yield from the vine, we got the vegetative growth of the vine. We know, we get an idea of the balance between those reproductive growth and vegetative growth. However, in our study, we found no difference among the yield, pruning weight, and also the ratio between them. So there's no difference. So again, because our vines are huge and they have very deep roots, and so they have those resources coming really deep down from the soil. So it is reasonable to not see those differences in a mature vineyard. And it basically confirms our previous study. So we also measure the grape basic composition parameters, such as Brix, pH, and total acidity, abbreviated as TA here. So in general, we did not see any difference, but in 2015, the grapevine cultivation treatments had a lower acidity. If you look more close into the Brix for 2015 cultivation here, you'll find that the number is really weird, because when in a normal maturation process, usually the Brix, the sugar content is coupled with the lower amount of acidity. So when you get higher sugar, you get lower acidity. But in this treatment, we found that the grapes from the cultivation in 2015 actually had a lower sugar and also lower acidity. It's very weird. So what we found is that in 2015, we had some vines in that treatment specifically had the leaf roll virus infection. So that's why the decouple of sugar and acids happened. And in 2016, a lot of those vines were naturally dead, or those vines that show the really severe symptoms, which is excluded from our experiment. So in 2016, we see that the number come back to normal. But basically, we did not see any difference among the treatments for those parameters. And also, we assess the sensory properties. I know a lot of you are very excited about this data, because a lot of you had contributed to this and got the money out of it. It's so the most important thing is you will no longer, do you remember those days when you open your email inbox and see like 10 emails from me asking you to do my sensory study? And do you remember that time when you see me in the hallway, like just sweaty and grumpy, trying to get people stuff in the room to smell my stinky wine? And I know that you're freaked out because I look like a drug dealer or something. But I guarantee you it's not going to happen again, because we got all the data here. But unfortunately, Annie is going to do that in the near future. So let's look at data. I'm going to explain to you how we did this. For those of you who didn't come to my sensory study, thank you, by the way. So we basically asked all the panelists to smell through all the wines and then sort the wine into groups based on their aroma similarity. So the wines that smells the same to the panelists, they just put it into the same group. And then we assign the score based on the grouping. And then we collect all the data, and then we calculate the distance matrix between or among the samples. And then we visualize it. So we visualize it based on the distance matrix. So I know it sounds very confusing and sounds very complicated, but what you need to know is that if the samples are closer together, that means their aroma similarity will higher. If the samples are further apart, that means their aroma similarity or lower. So as you can see in 2014, the glyphosate samples cluster together. So that means there's maybe a treatment effect, but let's look at 2015 and 16. So in 2015, now we see the clustering of cultivation, but not glyphosate. In 2016, we don't see any clustering or pattern at all. So which means we don't, like overall speaking, we don't see the treatment effect in each year of the wine sensory properties. So also measure the soil health parameters, including aggregate stability, organic matter content, microbial respiration rate, and carbon mineralizability. So here, in the third year of study in 2016, we basically see that the increase of aggregate stability and organic matter for the natural vegetation treatment. And it is very easy to understand because if you just let a weed to grow, that's the organic matter. That's organic matter. And during the winter, they got killed, and it's just a natural input of organic matter into the soil. So if you have a higher organic matter content, then you're expecting a better microbial activity and also a better aggregate stability. So it's easy to understand here. So in 2015, we also see an increase the microbial respiration rate in the natural vegetation, and also 2016 compared to the rest of the treatments. And so microbial respiration is an indicator for the microbial activity in soil. So, and it is important because, you know, if a higher microbial activity, that means you will have a higher nutrient mineralization in your soil, and it's beneficial to both your plants and the soil. So we see that natural vegetation increase the microbial activity there in both years. And for the carbon mineralizability, it's an indicator for the nutrient mineralization in the soil, and we found that natural vegetation increased the mineralizability, carbon mineralizability in 2015. So overall speaking, our soil health was improved throughout the years. So to summarize the physiological perspective, vine vegetative growth, fruit composition, basic fruit composition parameters, and wine sensory properties were not impacted. There are no difference among the treatments. However, natural vegetation treatments basically improve the soil health. So let's look at the result from our microbial study. So we sample the soil and the grapes in the vineyard. We grid sample 6 soil cores and 10 clusters from each of the experimental units. And then we combine all the subsamples for each of the experimental units. And then we extract the DNA out of it using those DNA isolation kits. And we are also interested to see the wine fermentation microbiome. So, so here I want to explain you very clear that we only want to capture the effect of the microbes. So we want to get rid of those great composition variation from different treatments. So what we did was we obtained a cornel juice from Lansing, that's a Riesling juice, and then we pasteurize it. So that's not coming from our experimental site, but it's just the base juice that we use, and we pasteurize it. We kill all the microbes in there, and we use the base juice to rinse 24 clusters from each of the treatment at our experimental site. So that means we dislodge those microbes on the surface of the grapes from each of the treatments, and then we use that to do our fermentation, to inoculate the base juice. So finally, during our fermentation, the only difference among the different batch of the wine, the only difference there is the microbiome difference. And the difference is coming from the treatment. So we really exclude that confounding factors of the berry composition difference among the treatments. And then we took samples throughout the fermentation, and the sample DNAs then sequenced used Illumina MySeq. And for bacteria, we target 16S region, and for fungi, we target IDS regions. So this is the result. Before I get into the result, I want to talk to you about the method we use. As you can see here, it says PCOA on the axis, which represents principal coordinates analysis. So what we did is we extract a matrix number from each of the sample, and that takes organism relative abundance, diversity, and the presence and absence of those organisms into account for each of the sample. And then among all those samples, there's a huge variation. So we extract 2 dimensions that explain the most variance among the sample, and we use those as our 2 axes. And then we plot the samples according to those 2 axes. Again, this is very complicated, I know. A lot of you are falling asleep. But just keep in mind that if you see the samples are further apart, that means their microbial community structure are less similarity. If they're close together, that means they're more similar to each other. So this is the soil bacterial community structure data for 2014. Basically, we can see a pattern here, that green dots, which is the natural vegetation on top of here. And then the cultivation samples are here. They separate according to the y-axis. In 2014, they just ran in together. It's hard to find a pattern here. But in 2016, we basically see that the blue dots, the blue samples, and the green samples, they separated. And these are visual result, but the visual result were validated with a paired Primanova analysis. So there's a number to actually show that your visual conception is true. So let's look at 2014. You see that natural vegetation treatments, the soil composition was very different from cultivation and glyphosate. So here. In 2015, there's no pattern, as you remember in the PCOA analysis. And yeah, it is true. There's no difference among the treatment. And in 2016, that natural vegetation, the green dots, is very different from the glyphosate, which is the blue dots. So the overall analysis also showed that natural vegetation really make the bacterial community structure very different from that of glyphosate and cultivation. And soil fungal community, we also analyze that. So there are two patterns you can you can find in the visual results here. So first of all, the round dot, those round dots are here, and all the square dots are here, and all the diamond dots are in the middle of the graph. So they basically cluster based on our x-axis, and the shape represents year. So basically, our samples cluster based on year. Year-to-year difference is important here. And also, you see there's a color clustering as well, because all the green dots, no matter it's dark green or light green, they basically separated. So you see that natural vegetation had a huge impact on the microbial community, on the fungal community structure here. And also this is validated by our paired Permanova analysis. As you can see, in 2015, 2016, and the overall analysis showed that the soil fungal community structure of each of the treatment being distinctive. So I want to remind you why we want to do this analysis. And also we look into the soil, and then we look into the grapes and wine, because many of you may have noticed that microbiome in the wine world has been a really hot topic. There's a lot of good papers coming out, and they all focus on one issue, that is microbial terroir. Microbial terroir is a very vague term, but basically they claim that microbes in the vineyard or microbes in the wine, they give the wine a sense of location. So we want to really look into that. And there are two leaders in the United States. They did a lot of good research on this topic, including Jack Gilbert, who came last year to give a talk. Jack Gilbert's from University of Chicago, and Dave Mills from UC Davis. So for Gilbert's group, they found that for vine aerial organ associated microbes, they're actually originated from the vineyard soil. So what's on the aerial parts, including grapes, is originated from the soil, in the vineyard soil. But they only did a bacterial community study. So we want to look into the fungi to see if that's true for fungi, because you know that it's very different for fungi and bacteria, and the spore size and the mobility is very different. So I want to see if that's true for fungal community as well. And for Dave Mills' group, they found a strong association among vineyard site, climate, and also the wine fermenting microbiome. So I want to study that as well. So later I will show you our wine data. So that's why we sampled from soil, grapes to the wine fermentation. So let's look at the grape data. We found that there's a very clear pattern. They cluster based on shape, but if you look into each of the cluster, there's no separation based on the color. So they cluster based on year. So year is an important factor here, but not color treatment. So treatment is not an important factor here. So that doesn't trigger the separation. Again, this data, the visual data, is being supported by paired Permanova analysis. As you can see that there's no significance among the treatments for all 3 years. So, which means, yes, adopting different under vine soil management practice really had a great impact on the soil microbial community, but this does not reflect on what's on the grapes. This is very different from the previous studies. So we take samples throughout the wine fermentation. So these are in 2014, and this is where we took the sample. So day 7 and day 9. So day 9 is the very beginning of the fermentation. As you can see, it's when the sugars start dropping. And day 13, which is a back slope, is at the peak of fermentation. Day 21 is a late stage, and day 48, that is at the end of fermentation. We took the samples throughout the fermentation process, and we want to compare the treatments here. So NI, non-inoculated, means it's just a fermentation control to see if the fermentation went wrong or not. So we actually what we want to compare is among these treatments here. So this is non-metric multidimensional sorting. It's a different method than the PICO-A, but the way to interpret the data is the same. So if you see the points are close, that means the microbial community are more similar, and vice versa. So if you see here, it's a very, very clear clustering pattern that the sample cluster based on day, the day of fermentation or the stage of fermentation, but not by treatment here. So what we see here is that the soil microbial community was heavily impacted by using different soil management practice. And then this does not reflect on the grapes, so nothing was spreading over into the wine fermentation. Another interesting thing that we found is this. So Saccharomyces is always being considered as the major player during the wine fermentation. What it means is during the wine fermentation, or at least at the peak of the fermentation, the Saccharomyces will present the most in the relative abundance because it's a major player, it's doing the work, it's actively growing. So the numbers of the Saccharomyces being the largest during the fermentation. However, in our fermentation, what we found is that Hansenia spora present the most in relative abundance throughout the fermentation and even at the peak of the fermentation. So if you look at the peak of fermentation, which is day 13, it's around 60% relative abundance, whereas the Saccharomyces is only at 15% of relative abundance. This is a huge difference. And so that's very different from what we normally think and from most of the previous study. However, we don't know what the actual function for the Hansenia spora is there in the wine fermentation. So just show you this very interesting results. Okay, so finally, the wine sensory study for the simulated spontaneous fermentation. So this is the results. So it's very, very random scattered of the samples on the two-dimensional plot. So basically, we did not see any patterns here. So it is very reasonable because we see no difference of microbial communities among the grapes, among the treatments, and also there's no difference among the treatments in the wine, so that the sensory properties were not impacted is very reasonable. And so, yeah, that's basically our microbial study. So to summarize it, we found that vintage being played the major role there in terms of the microbial community structure for soils and grapes. And natural vegetation impacted soil microbial community structure in terms of the bacteria and also the fungi. And fermentation stage was the only factor there to trigger the sample clustering on the two-dimensional sorting, but not the treatment. So the overall wine sensory properties were not impacted by our underlying soil treatments here. And in conclusion, for our the whole study, the underlying soil management did not impact on vine growth, yield, or the simple berry composition parameters of the mature Riesling vine and Finger Lakes. And natural vegetation generally improve the soil health. As you can see that the parameter we use, soil aggregation, organic matter, microbial respiration rate, and carbon mineralizability were all improved by using a natural vegetation. In terms of microbial community, that underlying soil management impact on the soil bacterial and fungal community. But that effect did not reflect on what's on the grape and what's in the wine fermentation. So overall speaking, there are no detection on the wine sensory properties, no matter it's through the conventional winemaking or it's through simulated spontaneous fermentation. So that's my study, and there's so many people I want to thank through my study here at Cornell. So firstly, I want to thank Sheldrake Point Winery because they provide me the land to use and to do the research, and they do everything they could to support us. And I want to thank Steve Lurch, our field technician and assistant, and the people at Geneva Brewing and Winemaking Lab, and also my previous and my current lab mates, like Adam Carroll is standing right there, and also my committee member, Anna Catherine Mansfield, Jenny Carnathan, Johannes Lehmann, and most important of all, my major advisor, Justine Venehuvel, who not only helped me through my research and also make me a much better person. Thank you. So I'm willing to take questions. We'll start from Geneva, and I will just... Okay, Geneva, do you have any questions?
Ming-Yi, just wonder if you can describe what your natural vegetation treatments look like. Were the weeds 3 feet tall, 6 feet tall? Was there any attempt to control or mow the weeds that were growing in this treatment?
Okay, so in 2014, because there's some the herbicide residues in the soil, so the weeds were not growing so well. However, they got like around 80 centimeter to a meter tall, and we didn't have to mow them down in 2014. But in 2015, they got really tall, so we had to mow them down like twice in the season. So like maximum that, for what I can remember, they can get to around my same height as me. So yeah, it can get really high, really tall for those, especially milkweeds. We saw a lot of milkweeds that get really tall in 2015. In 2016, they're not that aggressive, but we had to mow them down once in the very late season. Does that answer your question?
Yeah, thank you, Ming-Yi. Nice seminar.
Thank you. Thank you. Sorry. Do we go for Geneva first?
Sure.
Or? Okay. So please.
Really enjoyed your talk. I noticed in many of your analyses, years seemed very different. You might find a significant result in 2015 or the first year, but not the second. What do you attribute that kind of variation to? I know it was a drier year, but is there other confounding factors, or how do you make sense out of the big year differences you saw?
So yes, I will consider climate being the most important factor there, but there's a lot more than just climate. So if you look at different treatments, the weeds growing is really different among the year because of the climate, and also because of the disease pressure that different from year to year. So the spraying program are very different, and the way the vineyard crew managed the vineyard were different. But I will say the most important factor there is because of the climate that triggers the different weed pressure, different disease pressure. So that results in a very different data.
Thanks. Great talk.
Thank you.
Yeah, so I just wanted what, to what extent do you think there's any motivation then for mature, vineyard managers with mature vineyards to work towards improving soil health? You did say at the beginning, maybe less leaching of nutrients and pesticides. Would there be, to what extent are they fertilizing and save on losses? And what about soil compaction? Does that matter to them? Or just kind of trying to get a read of how a vineyard manager is going to respond to this?
Yeah, this is a very good question. So do I have to repeat? Okay, so the question is, what's the incentive for the growers to use different soil management practice because basically we see no difference. Is that
in yield or sensory quality?
Okay? Yeah.
Difference in inputs or environmental impacts.
Yeah. So if we think of it another way around, if there's no impact on the yield or the sensory property, why not doing something that is better for your soil, right? So if you adopt a natural vegetation and you're improving your soil health, that they give yourself more resilience to biotic or abiotic stress, then it's a good thing for yourself and for the long-term sustainability in terms of financial sustainability and also the environmental sustainability. And no matter what you do, you're not impacting on yield and also not impacting on the sensory property. So why not? So I think if there will be no like short-term turnover for doing those kind of practice. However, if you look at it in the long run, it is a good thing for not protecting your soil, also protecting a clean water, because if those the nutrients and pesticide leach into a waterway, that will get into our drinking water or into the lakes, and it's not good for those perspectives. Did I answer your question? Thank you. Great.
Ming-Yi, you mentioned the term microbial terroir, and so it seems like from your data, you disproved that hypothesis. So I'm wondering, is it worth going into that anymore, or do you kind of feel like it's been explored enough and it's really just something that sounds nice but doesn't exist?
So the thing is, I'm just testing in our region. So it is, I would say it's very region specific, because a previous study have shown that vineyard and climate is playing a major factor there. And also we proved that as well. So in our region specifically, what are the obstacles they're preventing the microbial terroir to happen is that we have a really, really big vine. So our vines are huge. So the spatial barrier from the soil to the grape is really far away. So even though we have a change in the microbial community in the soil, it is very hard. First of all, it's very hard to get into the fruiting zone compared to, let's say, in France, in Burgundy, in Champagne, they have, their vines are maybe 30, 40, 50 centimeter tall. So it's their cluster are just hanging next to the soil. And there have been studies showing that the mobility of the microbes in the soil to the air, it's about, for fungi especially, it's around 15 centimeter. So it's really difficult for us for the fungi in the soil to move a meter or a meter 20 centimeter tall to our fruiting zone. So that really prevents those to show up in our study. And also, as I mentioned, that we had very high disease pressure. So we had to spray so many times in a year. And even there's something getting to the fruiting zone, it was very hard for them to either thrive or to be able to be alive in the grapes, and then for us to capture it. So I would say in our specific region here, it is difficult to see, and there's those reasons, but it cannot be applied to everywhere in this world. It's really a region from region difference is contributing to that microbial terroir thing. Thank you.
So is the idea then that the microbes
they get on the outside of the grape, and that is as a result of dust, the field manipulations, the wind.
Yes, you have mentioned most of the reason, and also like the rain splash. There's studying being shown that the rain splash is playing a role there, and also the wind is playing a role there, and also like human as a vector to distribute is also very important.
So your study is really not about microbiome terroir, so maybe you could just define it so that people, you talked about Davy Mills and Jack Gilbert, which is great. So Davy Mills is really the most, more like microbiome terroir. So if you could just explain to people what it is, I think there's a lot of undergraduates that might be confused.
Okay, so microbial terroir is, let's define terroir first, and I try not to be too vague. Terroir is basically the sense of the place in the wine. That's what we call terroir. So you expect even the same grapes with the same winemaking techniques, people would expect it differently if you get a California Pinot Noir or a Finger Lake Pinot Noir. So that gives you a sense of place. So that's the term of terroir means. And microbial terroir is that what role does microbes play to give you that sense of place? So microbial terroir is basically that.
Thanks for the talk.
Thanks.
I was just wondering, did you look at all, you talked about spraying, I assume it was the same for all of your treatments, but did you look at that as an interaction term, like by year? I was just curious if you factored that in or thought that could be a confounding factor.
Yeah, I think it's worth looking at it, and we do have the spraying schedule and what kind of substance they use. But for the harvest, so a lot of data were analyzed at harvest, or the sample were taken at harvest. Before harvest is basically there's about a week of window without spraying, right? So basically it's the same. So I wouldn't say there would be so much of an interaction for that. Thanks.
One question.
Okay. Yeah. Okay. Sure.
Maybe go for it. Awesome, thank you. Thank you very much for the talk. So you were saying that the microbes travel via air. Is it not going through the root system? And is that another? That's why
that is the travel. Yeah. So her question is: does the microbes only travel through the air? Does it not travel through the plant? And the answer is yes, it does travel through the plant. So there's a lot of especially bacteria. There's a lot of them in the plants, and that triggers the plant or the vine and grape secondary metabolite synthesis. So yes, there's a lot of those, and I think that's important for our next step to see. So now we see that it's not impacting the wine sensory properties, and what is there to trigger the wine sensory differences in the very first study I showed. So we can really look into those endophytes to see if they are playing the trick here to really make the wine sensory properties different.
Right.
Thank you.
Thanks so much, Ming-Yi.
Thanks.
This has been a production of Cornell University, on the web at cornell.edu.